Hi, it's Kathy Deeks. I'm a little rusty on my technology, so I'm going to try to pull up this PowerPoint slideshow and review it. Seems like I should be able to share the screen. I don't know if you can see this. Share. Oh, that doesn't look like it's right. So, okay, so I'm going to talk about the mechanism of the AZT medication and how it impacts the HIV/AIDS virus for treatment. Just a quick overview, of the HIV life cycle. There are pretty well 6 stages that HIV would go through. And just to summarize these, we have the entry, where is the binding of the HIV to the receptors on the CD4 cell. We have reverse transcription, where the virus uses reverse transcriptase to convert its RNA into DNA. We have integration, where the virus uses the enzyme integrase to insert its viral DNA into the DNA of the host CD4 cell. We have replication, which is where the HIV replicates itself in activated cells and a proviral DNA is formed by transcription. We have assembly, where the new viral proteins and the enzyme move to the outer membrane. And then we have budding and maturation, where the virus bud is released from the host CD4 cell. So there are a few different drug classifications for treating HIV, and the one we're going to focus on here is AZT. And so next I'm going to describe that mechanism. So we have HIV RNA, and that it attaches to the reverse transcriptase enzyme and uses the host cell nucleotides to make DNA using the HIV RNA as the template. The viral RNA is knocked out. Then we have the reverse transcriptase, which sets up for integration into the nucleus. The HIV virus enters through the nuclear pores. There's the nucleus, an example of a nucleus, and here's kind of what those nuclear pores look like. And this is unique to HIV in that in replication, it doesn't need the cell to be replicating. It uses those pores. Then the integrase enzyme slices the new DNA into the host chromosome. And so now the viral DNA provirus is transcribed as a cellular gene. And our host RNA polymerase II is what's used to replicate it. Transcription of the gene produces a full-length RNA, leading to several proteins which bud out of the cell. And these are cleaved into functional proteins. They migrate to the cell membrane. And finally, we have the budding of the virion to occur, and viral protease cleaves and releases reverse transcriptase, forming the virion core. AZT is a thymidine analog, and on the far left here is the thymidine molecule, and in the middle picture is the AZT. They look a lot alike, and AZT works as a nucleoside reverse transcription inhibitor, and it undergoes intracellular phosphorylation by host enzymes, and the negative phosphates are added to the protein, And then, of course, through phosphorylation, we have the enzyme kinase, which transfers them to the target, changing the three-dimensional protein structure, and then, of course, its function. Reverse transcriptase would normally use the nucleotide analogs, A, T, G, or C, to build DNA strands, but AZT inhibits this as a competitive inhibitor. And then DNA elongation is terminated after the AZT triphosphate is incorporated. I found some amazing videos reviewing this with amazing production of animated videos that I can't create things like that when I just have a 6-hour day to put this together. And so I've linked those for you here, along with an excellent article from Up to Date that I had downloaded that overviewed all of the antiretroviral agents that are being used right now to treat HIV. Okay, thank you. Of course, I can't figure out how to stop this. So, okay, there we go.